All right, this video will serve to cover the second half of chapter 13, which we're going to cover in kind of a different way. Rather than talking about prions and then talking about the diseases caused by prions, we're actually going to talk first about the diseases and then circle back around and talk about the causative agent of those diseases, the prion. And the reason that we're going to do that is because prion diseases have been known to human beings for a lot longer than prions themselves had. In fact, the first spongiform encephalopathy was described in 500 BC by the Greek physician Hippocrates, who said, quote, "The truth of this is best shown by the cattle that are attacked by this disease, especially by the goats, which are the most common victims. If you cut open the head, you will find the brain moist, very full of dropsy, and of an evil odor." So he's the first record we have of a description, and he even went so far as to say that this disease, which occurred in cattle and sheep, he also believed occurred in human beings. Fast forward about 1,000 years, Publius Flavius Vegetius Renatus recorded cases of a disease with similar characteristics in the 4th and 5th centuries AD. And then finally, in the modern world, we see the first mention of this similar kind of disease in 1755, where an outbreak of scrapie was discussed in the British House of Commons. And during that description, they talk about the fact that they think that this disease had been present in Britain for some time, but it had come to epidemic proportions. And so there's a bunch of thought about what was going on. They didn't know if maybe it was contagious, what was happening to make it occur. And in general, they decided that it was due to inbreeding, and so they started making more careful, deliberate breeding choices for the sheep, and they appeared to be successful because the epidemic went away. And even until the early 20th century, there were no experiments that could show transmission of scrapie between animals unless they got super crazy and took some infected brain tissue and injected it into the eye of an unaffected animal. They couldn't replicate the disease. So they really didn't think that there was a link between whatever was going on in the organism and the ability for that to be transmitted infectiously through to another susceptible host. At the same time, they were trying to learn what these diseases were, and in 1921, a man by the name of Alphonse Maria Jacob described the spongiform encephalopathy, and what he characterized it by was what he called spongiform encephalopathy changes in the brain, which happened after a very long incubation time and resulted in multifocal spongiform changes, which is what you're seeing in this picture here, lots of different sponges, astrogliosis, neuronal loss, and very surprisingly to him and anyone else who knows about infectious diseases, there was no inflammatory reaction. So the body's first defense against infection was not brought into play against this spongiform encephalopathy, which again made people think this is probably not infectious. And in people, it started with maybe personality changes, depression, and then there'd be a lack of coordination and sort of an unsteady gait, or maybe involuntary jerking movements, insomnia, confusion, memory problems, and then patients tended to end up with mental impairment, dementia, basically, and exhibited something called akinetic mutism, which is a loss of the ability to move or speak. So this period of time was about 15 years from the start of these symptoms until the person died, because it always ended up being fatal, but there was really no idea of why it was happening, and it was so rare that there weren't a lot of people studying it. The first connection that people made between some kind of microbial pathogen and disease was the thought that perhaps there was some sort of very slow growing virus causing the illness, because they couldn't find any evidence of a pathogenic cell in the brain tissue of these people. And there are several of these kinds of spongiform encephalopathies. And the crazy thing is that they are not all caused in the same manner. You have sheep scrapie, which they thought to be a result of inbreeding, but it turned out you could inoculate, right? Remember I said you could, stab an animal in the eye with infected nervous tissue and get them to develop the disease. There's something called chronic wasting disease of deer, which again didn't have a clear infectious path. Bovine spongiform encephalopathy, which we call mad cow, kuru, and then this really interesting Creutzfeldt-Jakob disease, which is inherited apparently. So all over the map, people seeming to inherit a disease, people seeming to contract a disease, and people had a really hard time figuring it out. And then in 1982, a very provocative hypothesis was put forth by a man who had been studying a disease called kuru in Papua New Guinea. Still, nobody knew what was causing this disease until 1982, when a researcher, I don't remember his first name, last name, Prusner, coined the term prion for the etiologic agent and said, I think it's a protein, which blew everybody's mind, because how do you have a protein which is capable of causing disease? So this protein-only hypothesis met with lots of disdain. Nobody really thought that that was what was going on, but people started studying it, and it turns out that there is a protein that is normally found on chromosome 20. And when they discovered this protein, they called it the PRPC, or prion protein cellular protein. So I know it's a little bit confusing for them to call it a prion protein, right? So this is a normal cellular protein found in everybody's brain. In fact, found in the brains of not just human beings, but organisms throughout kingdom Animalia. And it's, like I said, found on chromosome 20. And it's thought that it's a protein possibly involved in long-term memory. People are not sure exactly what its function is, but when that normal cellular protein completely changes the way that it acts. And so what they found was that there were high levels of the same exact protein, amino acid sequence the same, but a very, very different three-dimensional structure. Within those plaques, there was a different form of the protein, which they called prion protein scrapie, right? So you can imagine they find prion protein scrapie first, because they're going into that plaque, because that's the unusual part of the brain, looking for what's inside it. And what they found is not empty space, as spongiform would suggest, but these huge, massive conglomerations of this protein, and they called it the prion protein, and they gave it the name prion protein scrapie, or you see their PRPSC, because they were naming that for scrapie, right? The disease of sheep that had been really well studied. So prion protein cellular has about 42% alpha helices and no beta pleated sheets, which you can see on the left-hand side of that diagram, whereas the scrapie form of the protein, once it's changed its folding, has about 43% beta pleated sheets and 30% alpha helices, which means it has a much more stable structure. And this change from the cellular form to the scrapie form seems to be sporadic. It can be a result of a sporadic mutation, and it can also be an inherited mutation. And in fact, it seems to be that you're higher at risk for prion disease if you are homozygous for methionine at a particular codon, as opposed to valine for that. So it can be sporadic, it can be inherited, It can be contracted iatrogenically, which means via a transplant, and you actually can contract it through the gastrointestinal tract because in some forms of this disease, these prions are actually found in the bloodstream, which is where we get that term prionemia. Emia means in the blood, so the presence of prions in the blood. Now that tells us where this prion is, but what does this prion do? Well, it turns out that the prion form of the scrapie form of the prion protein is something that the cell recognizes as something that needs to be gotten rid of. So, like it does with all of the other garbage it finds, it takes that scrapie form of the protein and it puts it in a lysosome, where under normal circumstances the protein would be digested and all of the amino acids would be freed up to be recycled for other uses. The problem, though, is that this scrapie form of the protein is so stable that it resists proteases, it resists radiation, nucleases, heat, alcohol, formalin, phenol. Right? It's really difficult to break down this protein so that it's not functional. So as the cell is trying to do the work of taking all of these prion protein scrapie forms and shove them into the lysosomes just get bigger and bigger and bigger and bigger until they crowd out and kill the cells. And it's really not known whether it's the crowding and the damage to the neurons which kills you, because this is absolutely a lethal disease, or is it that you just end up with a lack of the normal cellular protein? So not sure what exactly kills you, but there is no treatment for this disease, and all they can do is give you palliative care, where they're trying to relieve symptoms and keep you comfortable until the disease ultimately takes your life. So what we do know is that prions, though they can be transmitted, they're not transmitted through the air or through touching other forms of casual contact. They can be contracted if you have contact with infected tissue or body fluids or contaminated medical instruments. And we're going to learn in a later chapter what those different kinds of transmission are called, but this is something that is direct contact transmitted, and only when you have direct contact transmission between the infected body tissue or fluid from a person who has this scrapie protein, and that scrapie protein is allowed to find its way into your brain, where it can find those cellular proteins, because it turns out, right, you might be asking yourself, well, I mean, that's great, like we'll get a little bit of scrapie protein, but ultimately it'll all be packaged in those lysosomes. So why is it that people don't recover from this disease? At least some of them, right? They're able to package away those scrapie proteins and keep them from doing any damage. But the problem is that that scrapie protein becomes an enzyme when it takes on that scrapie form. And this was the part that people really had a difficult time understanding and believing, was the idea that this protein is self-replicating, because the scrapie protein can take the cellular protein and refold it into the scrapie form. And as I've said before, it's much easier to see these processes dynamically happening. So we're going to look at that in just a minute, but just really quickly to point out to you, so here comes the prion protein scrapie form, which looks like a little orange U at the top of the diagram. And notice in step 3, that scrapie protein grabs the cellular protein and changes its shape so that now there are 2 scrapie proteins, which we would call exponential growth. And then those 2 will turn 2 more, and now there are 4 scrapie proteins, and then 8, and then 16, and on and on and on and on. So as long as there is a single scrapie protein in the brain, every time the cell makes a cellular protein, the scrapie protein will convert it to scrapie. So it just becomes this huge problem, because your body is constantly making the hardware for the scrapie protein to be made. And the other problem with this is that, as I mentioned, these scrapie proteins are so difficult to kill. I mean, we'll use the word kill even though we don't kill proteins. The World Health Organization says that sterilization, which will actually kill the prions, is a much more difficult process than routine sterilization, right? Routine sterilization, 121 degrees Celsius for 15 minutes with a little bit of pressure and you're good to go. This one, you have to soak it in a prionicidal detergent, so a very special detergent that's able to start working on these prions. You have to immerse it in 1 normal sodium hydroxide for an hour. And then you have to sterilize for 30 minutes in a gravity displacement sterilizer at 121 degrees Celsius. Then you clean and routine sterilize. We're going to talk about a couple of prion diseases in the diversity unit. So what I'm going to leave you with here is just a video that shows you the replication of the prion.
All mammalian cells contain a gene that codes for the primary sequence of amino acids for the prion protein PRP. Normally, PRP folds into a functional form with several alpha helices called cellular PRP. The normal function of the prion protein is not well understood, but it's believed to be important in normal synaptic development and function, and it may be involved in stabilizing the structure of the synapses and in establishing memory. The prion protein is also capable of folding into a form with beta-pleated sheets. Prions do not reproduce like bacteria or viruses. Instead, infectious prions convert normal prions into more infectious prions by folding them into beta-pleated sheets. Thus, the number of infectious prions is increased, but by conversion rather than traditional reproduction. Prion proteins in the infectious conformation can group into multimers, which are very stable and resistant to protease. Over time, these multimers probably lead to some of the damage seen in prion-infected tissues.